All right. Hi, we're here at the Cold Spring Harbor Genome Stability and Integrity Meeting. I'm Krista Bledsoe. I'm an editor at Molecular Cell, and I'm here with Xiaolan Zhao, who has a lab at the Memorial Sloan Kettering Cancer Institute. Thanks for joining me today.
Thank you.
I know your lab works on the 3 Rs. Can you tell us a bit about what those are and what you work on?
Yeah. So the 3 R stands for DNA replication, recombination repair, and the DNA damage response. DNA replication is basically you accurately duplicate your genome, and that is a really difficult job because how many base pair our genome is and how many times we have to duplicate it so we can form a healthy individual. So we're interested in that process, particular from the angle how a replication fork, when it moves and when it's encounter barriers, or encounter transcription conflicts, the replication machinery know what to do, how to navigate through this journey of trouble, basically. And on the recombination repair, which is basically one of the most fundamental repair the cell need to have, not only just to fix a double-strand break, but also a lot of the replication gaps left behind by polymerase when polymerase doesn't know what to do. They will just run away, and then that gap left behind have to be taken care of by recombination repair. So in that way, these two processes are very intimately linked. The third R process we're studying is called DNA damage response. This is a system cell actually survey the genome to say, do we have normal basal level of DNA damage just going on spontaneously, or are we in trouble? Meaning we went to beach for so long, we get a lot of UV damage, and once the cell sense that increased level of damage, they need to reprogram a lot of the process to be in a stress state to cope with the emergency, and that process is complicated to study. And so the original paradigm is there is a checkpoint kinase mediated DNA damage response, and a few years ago we discovered a parallel pathway, which is SUMO mediated DNA damage response. Both are very important, and they collaborate. So we have been focused on mostly on this branch of the pathway to understand how cell use these two legs or two branch of the system to help it best cope with the stress. The other aspect we study on the DNA damage response is eventually after cell being stressed, they did a lot of things. For example, they would say, okay, we can't move on in cell cycle anymore. We are in trouble. Let's hold on the cell cycle progression and give more time to the repair. But once you do that for a while, you want to reverse that decision to say, look, it's time to move on, even we're not perfect yet. For example, we still have something not fixed, but we need to move on. Part of the reason is because if you don't move on, you don't have any chance to survive. That's the whole point, right? But if you move on, the next cell cycle phase, for example, G1, might provide you another repair opportunity that's not available in G2M, right? So we're trying to figure out how cell make that decision in terms of turning off checkpoint. Most people study how the checkpoint turned on or SUMO pathway turned on. It's much less investigated about how this process gets reversed. So those that area we study in this, I would say, interconnected arena on genome integrity. So regarding your question, why I'm excited about study this 3R, why I choose this particular field to study among many, many interesting questions. First, I have to say I am intrigued by many, many questions. Like this conference, we have so many interesting topics on development, and it just fascinates me to listen how the brain development. I have extra. If I have extra time, I will always tune in on this. But the reason I particularly choose this field, I think, is because it's so fundamental. When we talk about the brain development, in the end of the day, DNA or genome is still the command center, send out all these signals and messages to say development in this particular way, or die in this particular way, or transcribe. So the question is, regardless what you're studying, in the end of the day, the genome is still holds such important place in governing many processes. So that's why if I want to spend my limited lifetime to study something, I want to study something so fundamental and also have profound effect on other processes, hopefully, and so let people to think about the genome in different contexts.
Mm-hmm. Yeah, that's interesting. Yeah, we just heard a really great talk from you on the SMC56 complex. Can you tell us how that's different from cohesin and condensin, and a little bit about what you told us today?
Yes. So this is a very interesting complex. Actually, I purified or discovered this very serendipitously. I was long time ago was a postdoc with a lab, Gunter Blobel, wonderful PI, and the lab studied nuclear porins. So I actually did unbiased screen looking for any mutant would be sick with lacking nuclear porins, and that's how I identified this complex. So from its discovery, you already see it's very different from cohesin condensing. Cohesin condensing were discovered originally by their lack of cohesion or lack of condensation. So their name already reflect what they do. So in our case, we don't really know from our first encounter with it why it is synthetic with nuclear porins. So for a long time, we did not know, what to make of it. It's a really long journey because each field is sort of governed by a paradigm, and that paradigm has been established by cohesin condensing study. Beautiful, beautiful work. I was influenced heavily by that school of thought. But those type of models could not explain what we observe about SMC56 genetically. So I was trained as a geneticist, so we always have this belief the cell will tell us. Meaning when you make a perturbation, the phenotype would lead us to know what it does. But in this case, it's very complex. It's not straightforward. Cohesin condensing, beautiful, right? Mm-hmm. But we decided to look more in depth instead of superficially say, oh, the cell is sick, it's not too much cohesin condensing problem. What is it? So the field also spent a lot of time on characterize those defects. That helps us a lot. So zoom in 10 years, now we know better, but back step 10 years, we are also confused in field. So that some of the cohesin condensing scientists would say, this is a very confusing one. So they say, why don't you name it confusing? I thought also, it's funny name, but actually rhymes with cohesin condensing, but it reflects how, at the state, how little we know about it. Now zoom back 10 years to now, so many people have been worked very hard in different laboratories, studied this complex in different organisms. And you can see this is a highly conserved complex. Normally highly conserved complex has conserved roles. In this case, all the studies start to emerge and converge and tell us this is actually a specialized SMC. It's not like cohesin condensing like. What we know it have a direct role in DNA replication and repair. What cohesin condensing does is they organize chromosomes in different ways. Through that organization feature, it will influence replication recombination. But this complex goes directly to bind to the polymerases that does the DNA synthesis, binds to replication fork and enzyme that's manipulate the fork and tell them when to do that, when not to do that. So that was very satisfying for us because for a long time we don't have a handle about what exactly it could do. And but science always progress in such a way. Once you reach that stage, you have to say, but how does it do it? We know what it does do, but we don't know how. So that's where we decided to go back to the drawing board, ask very fundamental question. We don't even know how this complex look like. In early days when I was a postdoc, I remember the day when we mass spec this complex, find 8 subunits. We have no clue what they are. So we went back, tried to do some biochemistry to see how this complex, how this 8 protein come together, work as a team, as a complex. So the first step is try to get a lot of those protein, and that took many years to try. 8 subunits is not very easy to express in one cell, but we achieved that. So we're in good position. to assemble a team to help us to understand those fundamental questions, such as how this complex look like, at the structural level, how it binds DNA, what other activity it does. So my talk touched upon a little bit of each. The most simple summary is it acquired 3 extra subunits compared with cohesion condensing, and that acquisition helped this complex have more specialized activity. For example, the reason we think that this complex uniquely more specialized in replication repair is because one of those newly acquired subunits can target it to where those DNA sites are through a particular recognition. And the other subunit will help this complex to modify a lot of replication repair factor to make it more effective or maybe go to the right place. So, but there's still a lot more to learn, and we're very excited. And this is a teamwork. We have very good collaborators in our institute, the structural biologist Yu Yu from Deng Xiao Patel's lab, as well a former graduate student worked with us, Zheng Shi from Alex Kentis lab. My own postdoc, Shibai, play a very major role. They work very, very kindly with each other. It was a beautiful teamwork. I'm so excited every day. We, say, what's new? And looking forward, we're studying some more collaboration on biophysics of this complex with Shixin Liu's lab across street at Rockefeller. That's why our Tri-Institute play a major role in this, and our support for funding also very helpful. In our case, is funded by our internal fund just for our 2 labs, and we were, Sloan Kettering and Rockefeller also collaborate very closely with each other. So we have some very exciting results. Hopefully we can share with you very soon with Shixin's graduate student, MD PhD student Jeremy. I'll hopefully show you very soon about that data.
Yeah, that's a really neat story. Thanks. I wonder if, since you're working on structural biology, have things like AlphaFold or some of the single molecule techniques, have they changed the way that you do work in your lab?
That's such a great question, because I was trained as I was earlier mentioned, as a geneticist, but I always was very interested in utilize all the right approach to address the question. So when I was in Gunther's lab, I picked up biochemistry, cell biology. That's how I purified complex. But the landscape of those technology has been moving so much faster, and I was super excited to learn structural biology, single molecule through our collaborators. And to me, AlphaFold is such a revolutionized development, and we use them all the time because sometimes when you get a structure, you have a region of your structure not very high resolution, and the AlphaFold sometimes help us to improve the resolution in that area. And single molecule technique, one is by Lumic, several people mentioned about C-trap. We love that machine. It really makes it so much easier. Before you have to custom made your own machine. Now it was really beautiful, and we're using that technique as well.
Cool, thanks. Yeah, it was really nice talking to you. Thanks so much for telling us more about your research.
Thank you very much. I really enjoyed this meeting and meeting you, and a lot of colleagues, and learned a lot about new development. It's just such an exciting symposium.
Yeah, it's been a fun week. Thanks so much.
Thank you.